So, in meiosis, very similar to mitosis, but we're going to be talking about the key differences. In meiosis, you'll still have a lot of the things happening that we talked about with mitosis. Your spindle fibers form, cytoplasm is going to divide, but the chromosomes are the key difference. And that's because if you recall, we had mitosis takes one diploid cell with 46 and makes two diploid cells with 46. Whereas meiosis takes one diploid cell and produces 4 haploid cells. Okay, so that means that the chromosomes are going to be the big difference. So you still have your homologous chromosomes, okay, or homologs. Remember, they look alike. They're going to have the same size, length, genetic content. Their centromere position is going to be the same. Their DNA banding patterns are going to be the same, which means it's genetic content pretty much. Anyway, a specific location on one chromosome or one homologue is going to have the same gene occurring on the same location of its homologue. Okay, so if I have 2 of number 17, and number 17 contains a gene for eye color, then your first number 17 that you got from your mom might have blue eyes. The one you got from your dad might have brown eyes. So meiosis has 2 main divisions. Like I said, meiosis, it goes through PMAT twice. So that means 2 divisions. Okay, so it divides once into 2 cells and then again into 4 cells. So meiosis 1 begins with your diploid cell, and at the end of meiosis 2, you get 4 haploid sex cells. So, with meiosis, you still have interphase. You still need to copy your DNA. You need to double your DNA because you're still starting with a diploid cell with 46. Let's look at the math. When you double your DNA, you get to 92. So that will be an S phase of interphase, just like mitosis. And then in meiosis 1, you would get 2 cells with 46 each. And then after meiosis 2, they're going to divide again and get your 4 haploid sex cells. Okay, so that's how the math works out. So anyway, your chromosomes are going to pretty much still be the same because you've duplicated them. So you have your identical sister chromatids. They have the centromere in the middle. Remember, your homologous chromosomes are going to be similar in size, shape, and genetic content. So meiosis 1, you'll notice you have a 1 behind each of the phases. So it's still PMAT. It's still prophase, metaphase, anaphase, telophase. You just add a number to the end. Because like I said, you're going to go through PMAT twice. So meiosis 1, you're going to have a 1 behind PMAT. Meiosis 2, you'll have a 2 behind it. So this is a picture of meiosis 1. I would go as far as to say that meiosis 1 is much more important than meiosis 2. Meiosis 2 pretty much looks just like mitosis, except for you're going to end with cells that have less genetic content. It's going to have 23 chromosomes instead of 46. But in meiosis 1, you're going to have something very important happen, and it's called crossing over. That's going to happen in prophase 1. Okay, so if you look here, you can see the chromosomes are paired up, and they are actually crossing their legs. Okay, they're crossing their legs. I'm going to remove where I just highlighted. Okay, now what that ends up with is chromosomes that look like this. So look in metaphase. Notice how these chromosomes have both green and blue on them. So you have both green and blue. That's because of crossing over. We didn't show that in mitosis. Okay, so anyway, in metaphase 1, your chromosomes are still going to line up at the center, but there's going to be 2 rows of chromosomes instead of 1. In anaphase 1, your chromosomes will still be pulled to opposite sides of the cell, but instead of V's, they'll be still whole X's. Okay, and then in telophase 1, they are going to split and eventually become two new cells. They might not wholly split until they get to meiosis II. It just depends. But essentially, you've split out into two new nuclei. So, prophase 1, like I said, is arguably the most important step of meiosis. So, why is because of crossing over. You absolutely will have test questions about crossing over. Very possible that I'll put some sort of essay question about crossing over, because crossing over is the reason why humans are all genetically different and why sexually reproducing organisms are genetically variant. So, prophase 1. This is going to be meiosis 1, the very first step. So, first things first, we got to take care of that DNA, those chromosomes. So, first things first, you had interphase. You doubled your DNA, okay, and it was chromatin. So in prophase 1, your chromatin becomes chromosomes. And at that point, it is then visible. Your spindle fibers form. Your nucleus disappears, okay, and your centrioles are going to move to opposite sides of the cell, just like in mitosis. But something extra happens. The extra thing that happens is in step 2, okay. So you would say even that step 1 would be basically the same as mitosis. That's a weird S. But step 2 is the difference. So synapsis occurs. What synapsis is the pairing of your homologous chromosomes. Remember I said, like, if this is chromosome pair number 16, okay, you got one from your dad, one from your mom, well, they're going to synapse. Okay, so they're gonna match up and they're gonna pair up. When they're paired up, they're called a tetrad. Okay, so you have a tetrad. Then they cross over. Okay, so if you look here, you can see the blue and the pink. Your chromosomes from your mom and your dad have crossed over, and then in the resulting, at the end, you'll notice that these chromosomes have portions of both male and female. Okay, so from your mom and from your dad. So you're getting a little bit of genetic content from each parent. And this crossing over is going to happen differently every single time. So eventually, these chromosomes are going to go into 4 new cells. So the resulting baby could have this combination, this combination, this combination, this combination. Now remember, this is just the formation of the egg in one person's cells. You'll do the same thing with the formation of the sperm. Because this happens differently every single time, this is why humans are so genetically variant. This is why humans are so genetically diverse. Because this has happened so many times and we have so many chromosomes and genes, and this happens differently every time. This creates all the genetic variation that we have in our species. So crossing over is the exchange or swapping of genetic material. So it's the chromosomes basically swapping arms or legs. So like I said, this gives us genetic variation. So, and this is not just humans. This is going to be other animals too. The point where the arms cross is called the chiasmata. And this is where, like I said, the crossing over is actually going to happen. So it's where the arms cross over. Anyway, so I mentioned before, genetic recombination makes sure that your offspring are going to have different combinations of genes compared to your parents. And it's also compared to your siblings too. So there's 3 reasons that make you genetically different. The first reason is the chance or random combinations of haploid sex cells can give you all these different zygote configurations or even mutations possibly that are going to be new for the species. So crossing over is another one when crossing over happens, and then chromosomes are independently assorted. So that means they're lined up independently. This independent alignment causes the sex cells to have a different combination. So like I said, you might have more of your grandfathers or more of your grandmothers. So if you look at the math of this, in humans specifically, And, you know, different animals, different plants have different numbers of chromosomes. But in humans, you have 23 pairs of chromosomes, and then you have two parents. The combinations possible with one crossing over, this is just one crossing over. This is not counting both parents crossing over occurs in. Because when you make the egg, crossing over happens, and when you make a sperm, crossing over happens. So this is the combinations possible with at least one crossing over. It's going to give you this number. That's a very large number. Okay, so anyway, that's why you don't see people that are genetic clones unless they are identical twins. So fertilization provides us with different combinations of different genetically different sex cells. So that brings us to metaphase 1. In metaphase 1, your tetrads, same as mitosis, your chromosomes are moved by the spindle fibers to the center of the cell, to the equator. But the major difference is that they are lining up in pairs. Okay, so you will have not one, but 2 rows of chromosomes. Now you notice crossing over happened, so you have 2 colors in each chromosome to depict that you've got a little bit from each parent. So anyway, major difference is that they are lined up in pairs in metaphase 1. In anaphase 1, very similar to mitosis, the chromosomes are going to separate and move towards opposite ends of the cell. But another major difference is that the chromatids do not split like they do in mitosis. In mitosis, they split in the V's. They're V's moving to opposite sides of the cell. In anaphase 1, they're still whole X's. Notice they're still whole X's moving to opposite sides of the cell. So, it is important for you to make sure that you draw this correctly for your packet and your test. So telophase 1 is going to be very similar to telophase in mitosis. Your chromosome is still going to gather at opposite ends of the cell, and your cytoplasm may or may not divide. In telophase 1, you might have cytokinesis after it. You might go into an interkinesis afterwards. But anyway, all the normal things that happen in telophase are still going to happen. Those chromosomes are going to cluster towards opposite ends of the cell. So the resulting 2 cells have one homologous pair. And so these chromosomes are still whole chromosomes. They're still attached by their centromeres. So anyway, you need to divide one more time. So this is where your second division comes in, because at this point you have a diploid number. Okay, so you have 2 identical sets of sister chromatids. Well, in sex cells, we need a haploid number, so we got to divide one more time. So like I mentioned before, meiosis can occur a little bit differently. You might have cytokinesis, you might not. You might have interphase, or sometimes they call it interkinesis, which would be between meiosis 1 and 2. But you're not going to double your DNA at that point. Anyway, so meiosis 2 is similar to mitosis. If you draw it, it's very, very similar, except for you have 2 cells undergoing meiosis 2 simultaneously instead of 1. So in prophase 2, your new spindle forms around your chromosomes. And you may or may not have new nuclear membranes, so you might not have to deal with that part. Sometimes the nuclear membrane doesn't form back after meiosis 1. Anyway, in metaphase 2, your chromosomes are going to move to the equator, so they're going to move to the center of the cell again, and they will be in one row, just like mitosis. And then in anaphase 2, the chromosomes are separated and moved to opposite sides of the cell. So at that point, they are split into the 2 V's, so they're not whole X's anymore. So again, it looks just like mitosis. And then in telophase 2, which would be your last phase of meiosis 2 and meiosis altogether, your spindle fibers are going to go away because your chromosomes have reached opposite sides of the cell to their new location. Then you get 4 new nuclear membranes that appear, And then after that, you'll have a cytokinesis, which is going to divide out your cytoplasm into your 4 new cells. So if you take a look here, you have a picture of all the phases of meiosis 2 and 1, all of meiosis. So, I'm going to start out up here. We have our nucleus, nucleolus. You see a nucleolus present, that means that's interphase. Can't spell. Interphase. Okay, so then you have the cell. It goes into prophase. So both of these would be prophase. Okay, 1. Okay, so if you look here, you can see the DNA is still chromatin, but here it has become chromosomes, and they have paired up, and the tetrad is formed, and crossing over is going to happen. So it's very important you know crossing over happens in prophase 1. Okay, and then in metaphase 1, the chromosomes have lined up in 2 rows. Compare that to metaphase 2 and mitosis, which is just 1 row. Okay, so make sure you know the chromosomes line up in 2 rows in metaphase 1. Then in anaphase 1, they split. Okay, they're still whole X's. And the whole X's move to opposite sides of the cell. Then in telophase 1, you're going to see that the chromosomes are going to gather in the center of what's going to be a new cell. So they might divide and then go through another interphase where it grows, but they're not going to. If they go through another interphase, it does not replicate the DNA again at this point. We only do that in the first interphase. Anyway, so that brings us into prophase 2. So your chromosomes are going to cluster up in the center again. Your metaphase 2, notice there are 2 cells because we formed 2 cells. And so now those 2 cells have to go through meiosis 2. So metaphase 2, you're going to have 1 row of chromosomes. Then in anaphase 2, you have the V's. The X's are split into V's. Again, 2 cells. And then finally, you have telophase and cytokinesis, where you get your 4 haploid daughter cells. So at the end of meiosis 2, you have 4 haploid cells made from your original diploid cell. So if you look, mitotic cell division would be 1 diploid splitting into 2 diploids, whereas meiotic or meiosis would be 1 diploid splitting into 4 haploid. So remember, mitosis is going to be body cells and they're diploid. So you make 2 diploid body cells. Meiosis makes 4 haploid sex cells. Make sure you know that. During which phase of the process pictured below is DNA replicated? Well, hopefully you recall that DNA is replicated before meiosis, which would be interphase. So your best answer here would be prophase. You won't see that question on your test, though, because it's a bad question. But if you were to see it somewhere, you would know to pick the phase closest to it. All right, so fertilization. Remember, egg plus sperm gives you zygote. When the sperm fertilizes the egg, the zygote then has the diploid number. We've kind of went over and over that. Now, I mentioned before that zygote will then go through mitosis to become the multicellular organism that you are today. So you have your zygote, which then divides by mitosis, and then it becomes a morula, which is a bundle of cells. It's like a ball. So it doesn't look human yet. After that, you have what's called a blastocyst. Okay, so the blastocyst is going to continue dividing and growing, and you get more and more cells, and then it becomes a hollow ball. Okay, so we'll have, I didn't mean to hit that. then it will be a hollow ball. Okay, that hollow ball is going to have the amniotic fluid begin forming in the center of it. It's hollow because it has fluid in it. So that's where you'll get your amniotic sac with all the amniotic fluid. And it's a hollow ball because all the cells, you have to form the placenta. You're also forming the baby. And then there's also the yolk sac, which is going to give the nourishment to the embryo. And after it's in the blastocyst phase, then it becomes the embryo. So the embryo, it is the embryo until 8 weeks. And then it's considered a fetus. So the fetus is going to be until it's born, until the baby is born. And this is, of course, for humans. But it's very similar for like dogs and cats and pigs and other warm-blooded animals like us. Anyway, so when you're in the morula phase, the blastocyst phase, you're going to only have stem cells. Those stem cells are going to become any type of cell. And then when they get into the embryonic stage, you have tons of stem cells in the embryonic stage, but by the end of the embryonic stage, all those stem cells, well, not all of them, but a lot of those stem cells will have developed into all the organs. So all the major organ systems are developed after the embryonic stage when it becomes a fetus. Most people don't even tell other people that they're pregnant until after the embryonic stage. What's funny is because as I'm talking about this, my baby's kicking. Anyway, so cellular differentiation is when those stem cells become a liver or a heart or a brain or lung or liver cell. So I don't know if you know this or not, but the heart usually starts beating at around 6 weeks pregnant. This is my 4th pregnancy, and because of technology and ultrasound, I've been fortunate enough to see all of my baby's heartbeats at that time. Now the first 2 I did miscarry, so I only have one. I have one daughter right now, and then I'm pregnant with our son. But anyway, it develops very, very fast, very quickly. So it's very cool. Anyway, so the phases, you would have your zygote, morula, blastocyst, embryo, fetus. So this is just showing you kind of that development we were talking about. So if you look, we have the timeline off to the side here. Okay, so this would be just typical growth. So you start out with your fertilized egg. So that would be after the sperm and egg comes together. So if you look inside of the fertilized egg, what happens is that the sperm is going to enter the egg. Once the sperm enters the egg has this measure and it blocks any other sperm from getting in. Okay, so then if you notice here, you've got 2 circles. That's because you've got the sperm and the egg nucleuses, and they actually fuse. So at that point, your 23 and your 23 fuse to make your 46. So it is considered one cell, the zygote, the fertilized egg. And so it fuses to become diploid, the nuclei do. So anyway, then it goes through mitosis, and then you have 2 cells. Then you have 4, 8, so on and so forth, exponential growth. And so very quickly, these cells are going to divide. So day 1, you already have 2 cells. Day 3, you already have between 6 and 8 cells. This is before it's even implanted into the uterus yet, mind you. Day 5, you went from the blastocyst to the morula to the blastocyst. Sorry, I said it backwards. And so if you look at the blastocyst, you'll see that, it's getting the fluid. See the fluid in the center? Okay, so what you're seeing is you're seeing the placenta developing around. So you're getting that placenta to form that the blood so that the baby can get blood from the mom. And then you've got your fluid-filled area, and then you have what's going to be the baby. So anyway, all this, so day 7 is average time for implantation. So all this has happened probably before it's even implanted into the uterus. Again, you would not even know you were pregnant. Pregnancy tests cannot detect pregnancy until a couple of days, several days actually, after it implants into the uterus. It has to implant deep enough to connect with the mother's blood supply. Anyway, so this is showing you some pictures of, this is considered post-ovulatory days. So, if your typical cycle is 28 days, so if you have a 28-day cycle, then you would have, this would be the day of your missed period. This is what it would look like. This would be 13 days past ovulation if you had a 28-day cycle. So this is when you would find out at the earliest that you were pregnant. Anyway, so as you go, so that would be like 4 weeks pregnant. So if you just do the math on this, you can see all the different days past ovulation. So if you come up here, you have 56 days past ovulation. So 56 days, that's about 2 months. So this is going to be around 8 weeks pregnant. So around 8 weeks pregnant, you have a very, very, very humanoid looking baby. It's really cool. So anyway, you can see the heart forming here. Okay, so 41 days after from your previous period. So 17 days. This will be around 5 weeks. You're getting that heart. I've seen the heartbeat as early as 5 weeks, 4 days personally. So you're seeing that heart form, and it will start beating. And then as the baby gets bigger, and of course these are different pictures, you'll get more skin around it too. Anyway. So differentiation, again, that's when stem cells become all the other cells in your body. So that baby will become, or that embryo will develop the muscle, the blood, the nerve, and all the other cells. So that brings us to comparing. We like to compare and contrast things in science. So if you're comparing and contrasting mitosis and meiosis, then they have some things in common. DNA replication is still going to only happen once during both. Okay, but in meiosis, you have 2 divisions. You have 1 division in mitosis. So meiosis has 2, mitosis has 1 division, and that's how many times it goes through PMAT. In mitosis, you make 2 daughter cells. Meiosis, you make 4. In meiosis, they are gametes, which are sex cells, and they are haploid. In mitosis, they are diploid, and they are somatic cells. Okay, so another major difference is in meiosis, they are not genetically identical. They are genetically variant or genetically different. So meiosis or meiosis makes genetically different cells, whereas mitosis, they are genetically identical to each other and to the parent cell. Okay, we have 2 questions on this slide. Human cells have a total of 46 chromosomes in 23 pairs. Which of the following represents the changes in chromosome number through the stages of meiosis or meiosis? Okay, look at what it starts with, look at what it ends with. Well, hopefully you recall that meiosis starts with a diploid cell, which is 46, so I would eliminate A and D. Hopefully you also recall That meiosis makes 4, 1, 2, 3, 4 haploid cells. So C should be your answer. Your second question, which of the following is a characteristic of asexual reproduction? Offspring are identical to a single parent, unique haploid gametes, unique offspring from two parents, or crossing over during synapsis. Well, hopefully you know that all of these are sexual because they all apply to meiosis. Whereas asexual, you're going to have identical offspring from one parent. Meiosis is different from mitosis because meiosis produces consistent genetic makeup of all gametes, larger daughter cells, 2 gametes for every original parent cell, cells with half the number of chromosomes. So which one does meiosis make? Hopefully you picked the last one, haploid, cells with half the number of chromosomes. You do not get 2 gametes, you get 4. They're not necessarily larger daughter cells, and they are not consistent genetic makeup. They're going to be a variant or different genetic makeup. Saying consistent genetic makeup is like saying identical. So you are very likely to see questions like this. The number of chromosomes in the egg of a potato plant is 24. The number of chromosomes in the cells of the potato root is how many? Okay, so hopefully you know eggs are what kind of cell? Sex cells. And so sex cells, are they diploid or haploid? Haploid. So 24 is our haploid number. So then you have to look at the potato root. Okay, so a root is going to be a somatic cell. And we know somatic cells are diploid. So if the haploid, they gave me the haploid number, which was 24. So what would the diploid number be? That would be 48. Similar situation. A species with 12 chromosomes in each somatic cell will produce sex cells with how many? Okay, so if it's 12 in the somatic cell, we know somatic cells are going to be diploid. We also know that sex cells are haploid. So it gave me the diploid, and it wants to know the haploid. So hopefully you picked 6. What is the primary difference between mitosis and meiosis? Mitosis involves 2 cell divisions, and meiosis involves only 1. Mitosis produces diploid cells. Meiosis produces haploid cells. Interphase and DNA replication occur only in mitosis, or there's no difference between the two processes. Well, hopefully you picked this one. Mitosis makes diploid, meiosis makes haploid, because it's the only statement that is true. So that brings us to a change in chromosome number. How do we see that? I mentioned that was possible before, but now we're going to talk about how we can see that. So that's going to be through a karyotype. So a karyotype is basically a picture of a person's chromosomes. Okay, so it's a photograph of your chromosomes, and it's going to occur from a somatic cell. So it's going to be diploid. You're going to have 46 chromosomes, and they'll be lined up in 23 pair. They are arranged by size and shape. Okay, they're put in order. And the reason why you would do a karyotype is to check for a chromosomal abnormality. So maybe they have too many chromosomes, not enough chromosomes, or maybe there's a mutation where you're missing part of it, or maybe you have too many, whatever. A karyotype is very helpful in diagnosing things like Down syndrome. Down syndrome is one of the most common diseases or genetic conditions where you have an abnormal number of chromosomes. So with a karyotype, your chromosomes are paired up by their homologues. Their homologous chromosomes are paired up. And so you can look at the number of chromosomes. So here is a picture of a male and female karyotype. Okay, so if you take a look here, you will notice a few things. So first off, you have, they are numbered. Okay, and then you have your 23rd pair, which is your sex chromosomes. So if you notice your XX, they look the same, but your XY looks different. That'll be important for when we get into genetics later on. But anyway, so those are your 23rd pair, your sex chromosome. All the rest are your autosomes. So notice you have 1, 2 of number 1. There's your pair. 1, 2 of number 2. There's your pair. 1, 2 of number 3. There's your pair. Do you get the picture? Notice how they're arranged by size and shape. These are very small. These are much larger. And so they're arranged from largest to smallest. And this is based on chromosome number. So I can very easily see if this female or this male had an extra chromosome or was missing a chromosome, and I can also see if it's a boy or a girl. So, anyway, your chromosomes, of course, are your DNA. So they are super important in, analyzing and looking at humans. So anyway, each of your chromosomes has tons of genes on it. So one chromosome's got tons of genes on it. And all your genes help determine how you develop, how you look, and even how your body functions. The presence of all 46 chromosomes is essential. That should say essential. For normal development and functioning, if you're missing one chromosome, you usually will not survive. The zygote would not survive. You would probably miscarry the baby. Or even if you have an extra, many times it would be miscarried. Okay, so if you take a look at this karyotype here, you'll notice that you got the box here. You have 3 on number 21. So this individual is going to have 47 instead of 46 chromosomes. And it is a male. So you have a male with trisomy, tri for 3, somy for chromosomes, 21, which is Down syndrome. So anyway, they could easily do this and see if you had a chromosomal abnormality. So this is just putting it into writing what I just showed you. A karyotype is helpful in diagnosing Down syndrome. So again, someone with Down syndrome will have 47 chromosomes. So they will have 3 chromosome number 21s. Okay, so you can tell if you have an extra copy of number 21, or maybe if you're missing a chromosome, or if you have 3 X's instead of just 2. But what a karyotype cannot do is it cannot show genes. It can only show the whole chromosome. And remember, I said chromosomes have tons of genes on them. So anyway, how they get a karyotype? Well, you take body cells, somatic cells, and you look at them under the microscope during a stage of mitosis where they're most visible. So that's usually metaphase. And so they stain them. They stain the cells, and then they're going to be photographed. Anyway, so again, they're going to be arranged based on a standard numbering system from 1 to 22, and then your 23rd pair is going to be your sex chromosomes. So it's going to reveal any gross. Gross would be like major chromosomal abnormalities. So my husband and I actually had a karyotype done of both of us. As I mentioned before, we had 2 miscarriages before we had our daughter. And after the second miscarriage, it's miscarriages are actually more common than you think. People just don't talk about it because it's very, 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 it's a hard process to go through. I talk about it because I feel like when you get older, you're going to know someone that goes through that. Both of my sister-in-laws have had miscarriages. I've had a miscarriage. Most of my friends have had miscarriages at some point. Not all of them, fortunately, but I know many people who've had them. And it's just not something that people talk about a lot because it is very upsetting, obviously. So anyway, how you would get extra chromosomes or not enough chromosomes is because meiosis didn't go just right. So what's going to happen is, specifically like for a trisomy or a monosomy would be 1, having 1 chromosome instead of 2. is the chromosomes do not separate. Okay, so if you recall in metaphase 1, they line up in the middle in pairs. Well, in anaphase, they're supposed to separate. Okay, so this chromosome should have went over here, but it didn't. And when it split into 4 new cells, you ended up with a cell with too many and a cell with not enough. And depending on which one gets fertilized, then that baby could potentially have one of these conditions. Now, these are some known monosomies and trisomies that can survive. And I say survive because they might have some medical problems. People with Down syndrome, my uncle has Down syndrome, so he does have some medical problems. He is 50 years old, which is considered very, very, very old for someone with Down syndrome. We've taken very good care of him. He actually had a stroke about a year ago, and he's very lucky to be alive from that. But he's happy as can be, and we're happy that he made it through. But anyway, some of these syndromes are going to have very severe problems. They might have heart defects. They might have something wrong with their liver. They have lots of medical problems usually. So anyway, here's just some examples. Down syndrome, of course, is going to be the most common. Cat-eye syndrome, Cri-du-Chat, Turner syndrome is going to be specifically of the sex chromosomes. And when we get into genetics, we'll talk more about those. So anyway, non-disjunction. So you have metaphase, it lines up in the middle. And then in anaphase, it's supposed to split. And it doesn't split appropriately. So when those split into 4 new cells, this one, if it's fertilized, will become a trisomy. This one, if it's fertilized, will become a monosomy. If these two are fertilized, they will be normal. So it just depends on what the egg and sperm, which egg and which sperm comes together, if they have any of these conditions. Now, non-disjunction is going to be much more likely the older the mother gets. It can happen in egg or sperm, but it's more common the older the mother gets. And age 35 is the kind of magic number. At that age, your risk becomes much higher. Now, if you're older than 35 when you have kids, that doesn't mean you're going to have a child with one of these disorders. It just means your risk is higher. The percentage is higher. I know lots of people that have had kids between 35 and 40, and they've all been totally normal. So anyway, again, this is a more digitized version of a karyotype. So I can look at this and identify that this is a male because it's XY, and that they have a trisomy on number 21, which is Down syndrome. So again, my husband and I had karyotypes done. I actually have slides of my blood and my husband's blood. So, I'm able to look at them under a microscope, and I do share that with my classes, if, time permitting. And then I also have printed copies of our karyotypes. I'll try to remember to load a video on of that. But anyway, I show my classes my karyotypes every year, and I always ask if they know which one is me and which one is Mr. Bell. And it's funny because they don't look at the chromosomes, they look at our names at the top because it has my name or his name, rather than actually looking at the chromosomes to see who's XX and who is XY. Anyway, so this is a karyotype. You might be given a picture of a karyotype and be asked to identify if it's a boy or girl, or if you see any chromosomal abnormalities, and just know you'll look the last pair to see if it's a boy or girl, XX girl, XY boy, and then you'll check to make sure that you have 2 of each pair. And again, we get down here and we have 3, so that's Down syndrome. The purpose of meiosis is production of haploid gametes, asexual reproduction of unicellular organisms, duplication of diploid cells, or duplication of diploid gametes. Well, hopefully you know that meiosis makes haploid gametes. The final products of meiosis are like we just said, haploid. Meiosis results in greater genetic variation than asexual reproduction because it and hopefully you recall, we talked a lot about this, it allows for recombination of genetic information. Compared to sexual reproduction, asexual reproduction results in, so it's asking about asexual, do you get greater variation in offspring, larger number of identical cells, longer lifespan of a cell, or fewer number of cells dividing? Hopefully you know asexual gives you a large number of clones, identical cells. What are two sources of genetic variation associated with sexual reproduction? Is it binary fission and budding, mitosis and meiosis, crossing over and independent assortment, or diploid gametes and DNA replication? Well, binary fission and budding are both asexual. And this is sexual and genetic variation. So where does the genetic variation come from? Okay, mitosis is asexual. Okay, so meiosis is a type of sexual reproduction, but it's asking for the sources of genetic variation in sexual reproduction, which is meiosis. Are gametes diploid? No, they're haploid. Okay, so it is crossing over. Remember, that's when the genetics, your chromosomes switch legs, so you're swapping genetic information. And then the other part is independent assortment, and that's how the chromosomes line up independently of each other. Bacteria cells reproduce by binary fission, a type of asexual cell division. One advantage of binary fission is greater genetic variation in daughter cells, the ability to reproduce quickly, greater resistance to disease, or the ability to live under anaerobic conditions. Well, hopefully you recall that asexual can reproduce very fast. The primary difference between sexual and asexual reproduction is asexual reproduction occurs only in multicellular organisms. Asexual reproduction requires a multicellular parent and a single-celled parent. It's a typo, sorry. Sexual reproduction combines DNA from parent cells, or sexual reproduction occurs in only single-celled organism. Okay, hopefully you know that sexual reproduction is combining the DNA from two parents. According to the graph, one reason that more of the animal A population survived is because sexual reproduction produces what? Individual variation within a population, larger numbers of viable offspring, more females than males, or different forms for each generation. We'll recall that genetic variation allows us to survive better. So individual variation is saying that you are different than I am. So what kills you might be different than what kills me.